My name is Christine Morales, and in this video, I am going to answer questions pertaining to subjects learned during Unit 2 in Chemistry 1005, also known as Medical Biochemistry 1. So the first question I picked is Case 2, the regulation of glucose uptake by the cell. Here we see an image that depicts an MRI of an individual with a large glioblastoma tumor. So the tumor is actually these white areas on the left side of the image. And on the right, we see a PET scan of the same individual after they were given fluorescently labeled D-glucose. This again is still the brain on the PET scan. So I am going to use the following prompts to determine if and how glucose uptake differs by comparing the tumor to the surrounding tissues. We can see in the image that the tumor takes up significantly more glucose since it is more lit up than surrounding tissues, which are significantly darker. First off, I'm going to talk about how glucose uptake is regulated in the liver, skeletal muscle, brain, and adipose tissue. In the liver, the primary glucose transporter is GLUT2, and it is taken up in an insulin-independent manner. Skeletal muscle uses GLUT4 in an insulin-dependent manner. Adipose tissue actually does the same, GLUT4, insulin-dependent manner. And then the brain is different. The brain actually has two glucose transporters, and those are GLUTs1 and 3 in an insulin-independent manner, much like the liver. Now, when I say insulin independent, it means that the glucose transporter is always on the cell membrane. It doesn't matter whether insulin is there or not. So in this case, that would apply to the liver and to the brain. When I say insulin dependent, that means that glucose is only taken up when insulin is present, like in the case of skeletal muscle and adipose tissue. Now, it's clear to see from the image that glucose uptake in the tumor cells differs from that of normal cells. The tumor cells need a lot more energy since they have increased proliferation. They achieve this by taking in a lot more glucose than normal cells. So the cancer cells do this by something called the Warburg effect. This is when the cancer cells actually make the choice to use glycolysis, even when oxygen is present, instead of oxidative phosphorylation. Glycolysis is actually less efficient at producing ATP. With glycolysis, they only yield 2 ATP, as opposed to 36 ATP from oxphos. But this is still advantageous to them, because the cells can compensate for the loss of ATP by upregulating genes that encode glucose transporters and glycolytic enzymes, causing both an uptake in glucose and an altered metabolism. This change in their metabolic state allows them to survive even in hypoxic environments, which means not enough oxygen. This could be advantageous in the case of a large glioblastoma tumor, because as you can see, the tumor is basically pressing up against the walls of the skull, causing the brain to squish, causing the capillaries to compress, not allowing blood to flow to that area of the brain. That means not enough oxygen is also getting to that area of the brain. But despite no blood supply and thus no oxygen, the brain, to that part of the brain, the tumor is still able to survive. In fact, the inner parts of the tumor are actually also hypoxic, but this actually causes an advantageous effect for the tumor. It allows for a cascade to happen that causes an increase in GLUT1 expression so that more glucose can cross the blood-brain barrier to the tumor. This again allows the cells to shift to glycolysis as their primary source of ATP synthesis, which is advantageous to the invasive tumor because the cancer cells have the ability to use glucose at a higher rate than normal cells and can use a smaller amount for oxidative phosphorylation. Also, this altered metabolic state allows the cells to have more biosynthetic building blocks, like the intermediates and substrates for the synthesis of nucleotides, proteins, and cell membrane components, which can be used by a proliferating cell. Furthermore, This allows the cancer cells to use more glucose metabolites in macromolecule synthesis instead of carbon dioxide production. So the cancer cells may have changed from an M1 isoform to an M2 isoform, specifically a low energy state, so that they can alter glucose uptake. The low activity state of an M2 isoform allows for the production of glycolytic intermediates to enter branches of the glycolysis pathway, making NADPH, so that reactive oxygen species production is suppressed. So my second question is from case 4. And firstly, it's important to note that the baby's lab results are consistent with hypoglycemia, and that when epinephrine is administered, there was an increase in blood lactate. Results also show that the baby suffered from hepatomegaly and enlarged liver, and that she can't maintain normal levels after fasting for blood glucose, but that her levels did increase after breastfeeding. Now, hepatomegaly does occur with all of these choices, but it should be noted that in the case of the glucose 6-phosphate dehydrogenase deficiency, splenomegaly is more common than hepatomegaly. I will say that I am able to rule out galactosemia immediately because galactosemia is caused by a deficiency in galactose 1-phosphate uridyl transferase. This doesn't allow galactose to be changed into glucose. This deficiency is also known as a GALT deficiency, G-A-L-T. So the baby has been breastfeeding since birth, has not had anything else other than breast milk since birth. Galactose is present in the breast milk. If the child had galactosemia, they would have presented with vomiting and diarrhea immediately after their first feeding, like when they were born. And later in a few weeks, hypotonia, which is low muscle tone, jaundice, and hepatomegaly. That's why I'm able to rule out answer B immediately. In the case of hereditary fructose intolerance 1, I assume that they mean the one caused by a deficiency in fructose 1,6-bisphosphate aldolase. So the infant would have been asymptomatic unless they would have had fructose or sucrose at some point in their life. So the sucrose is normally from table sugar, and fructose would be from things like fruit, sweetened cereal, or sucrose-containing formula. But since the child has only had breast milk since birth, we can cross this out as well. That leaves us with glycogen storage disease and glucose 6-phosphate dehydrogenase deficiency. A deficiency in glucose 6-phosphate is often associated with causing hemolytic anemia, which the baby does not have any symptoms of. This is needed to make NADPH, which will then regenerate GSH when it is oxidized by a radioactive oxygen species. But if NADPH production decreases or is limited, then the cells could undergo oxidative stress, symptoms of which include jaundice, discoloration of urine, splenomegaly, and then sometimes hepatomegaly. Again, as mentioned before, hepatomegaly was part of the symptoms that the baby presented with. The other symptoms are not consistent with the findings, so we are going to go back to that and instead jump to glycogen storage disease. I'm going to use glycogen storage disease type 1 as the example, and those who present with this tend to present with fasting hypoglycemia. The baby did that. Lactic acidosis, there was an increase in blood lactate, and hepatomegaly. So type 1 glycogen storage disease is caused by glucose 6-phosphatase, or a G6P deficiency in the liver, kidneys, and intestinal mucosa, which would then cause an inadequate conversion of G6P to glucose in the liver. So I'm pretty sure that that the correct answer to case 4 would be glycogen storage disease, since all the symptoms that are associated with it were present in the child. So I am also going to rule out choice D.